I want to introduce Dr. George Church. He is the father of gene editing. On everyone's lips is CRISPR. Don't ask me to say what CRISPR really stands for. That's why Dr. Church is here. His biography is on the app. Please, Dr. Church, come right up and thank you. And Dr. Coleman, you've got plenty of time later, so don't worry about that. Let's give a hand for Dr. Church. Thank you.
Well, I won't tell you what CRISPR means either, in terms of an acronym, but I will tell you what it means in terms of technology. And I was planning on cutting my 30-minute talk to 9 minutes, but maybe I'll compromise on that. If I can have the first slide, it seems to be up on the... Anyway, it's wonderful to be here. I have to reflect and amplify exactly what the previous speakers today have said of how wonderful this community is. I'm going to jump right into where the outline of this 9 to 30-minute talk, which is talking about causality and how we can use stem cells and CRISPR to get at causality and preventions. How we can go beyond one genome change per cell. There's a tendency to talk about this as gene editing, and I'm talking about genome editing. And then how that genome engineering specificity is achieved, how we can do not just genomic but epigenomic engineering, and then how to analyze those. In terms of causality, as a genomics person, I'm surrounded by statisticians and myself who look for large cohorts, but every now and then, very often, the real nuggets come from N=1 studies where you have one individual who is unique and you don't have repeats, but you can nevertheless go from correlation, which in a certain sense is weaker even with large cohorts, to causality. And here's an example of a child who was born as a myostatin double null, and of course all that you knew was the striking phenotype and the genomic changes. And then you can confirm that with either animal models, such as here, 3 different animals having myostatin double nulls, or you can do it in human. For human, we have emerging human genome and cell standards, and I urge you to take these very seriously, as the National Institute of Standard Technology and the Food and Drug Administration have done in establishing their Genome in a Bottle project in collaboration with our program called personalgenomes.org, a nonprofit that provides the world's only source of such cell and genome standards. And these include not just stem cells, but other synthetic biology resources such as bacterial artificial chromosomes, 150 kilobase pieces that can be used for genome engineering, as well as CRISPR resources and complete genomes. And this is now international. There are 5 announced centers and 20 in the queue worldwide that provide this genes, environments, and traits for humans. And here's some examples of some of the stem cells and the teratomas and other complex tissues that have been made, as well as whole organism assays such as this fMRI. Now, when we move increasingly to human testing via organoids or organs on chips, they initially started with rather poor choices of cells, but very pragmatic ones, and we increasingly are going to epigenetic reprogramming of the cell standards that we talked about before from the Personal Genome Project. This is done in collaboration with Don Ingber and Kit Parker at the Wyss Institute for Biologically Inspired Engineering in Boston and Cambridge. And I won't go, there's a variety of different tissues that have been established in this, and there's a device that connects these 12 different tissue types and allows microscopic and chemical analyses. If we move up from organs on chips, very monolayers but more complex mechanical, to organoids, which are even larger, we run into challenges with vascularization, where you should have a capillary every 40 microns or so. And we have these various approaches: developmental biology, self-assembly, 3D printing, scaffolding, and even humanized animal contributions. And the point of this is not just to test the hypotheses, but test therapies of various sorts and improve the quality. So what about specificity? Now, the CRISPR-Cas9 system can be compared to previous genome and gene engineering and library efforts, including RNAi and cDNA delivery. RNAi is typically limited to having, it has some off-target issues, and it also is hard to get more than a 5-fold repression. In contrast, the CRISPR-Cas9 can go essentially to 0 via deletions, but it also has activation and repression, activation up to 20,000-fold. We've seen in quite a number of cases. And so this diagram shows the native CRISPR, which causes double-strand break on binding an RNA guide. So just 20 nucleotides, in fact, an improvement of 17 nucleotide region corresponding to the genome causes this triple helix and a double-strand break. You can knock out one of those cleavage catalytic parts of the Cas9 protein and get a nick, and you can put 2 nickases together to increase the specificity, and this can improve specificity up to 1,000-fold, as we'll see in a moment. And then finally, you can make epigenetic changes with activators and repressors. Now, why would we want nearly perfect specificities? We have this quite good specificity, less than 1 error in about 10 to the 11th base pairs. So with the initial system, the original system we published in 2013. But we would like to be able to do surveillance functions. We'd like to be able to systemic delivery to all of the cells in our body, or a large number of them. And when you start delivering to a large number of cells, you have to have a very low error rate. And we want cases where we want to go beyond the library, where you put in one genetic change per cell. You might want to make multiple changes per cell, as exemplified by the IPS protocol that I'm sure you're all familiar with, where you need 4 gene activity changes, 4 genomic changes essentially, to get the right answer. And we think that's going to be increasingly common in the epigenetic reprogramming. Here are some of the 4 of the ways that we have improved the nuclease specificity. We think this is poised to be possibly one of the most specific pharmaceuticals in history. All of them, small molecule drugs or RNAi or monoclonal antibodies, they all have off-target, but these ones we've seen in the very short, little less than 2-year time since this became a technology, 1,000-fold improvement via theoretical and empirical searches, 100-fold, 1,000-fold for the paired nickases, up to 500-fold. It's a little more complicated than that for truncated guides. We reduce it from 20 to 17 base sequence recognition, and about 140-fold for the paired nucleases that the Jung and Liu labs introduced. I'm going to skip over these 2 slides because we're going to have a session at 10:30, a longer session, and get to the other aspect of specificity. Those are off-target, where the CRISPR will bind to a region far from the target, and another problem is when it binds to the target, but instead of doing exactly what you want, it makes a mess, sometimes called non-homologous end joining, which can introduce insertions and deletions. If your goal is to get homologous recombination, even being on target is not enough. You have to also increase the homologous recombination to non-homologous end joining ratio. Now, there have been at least 15 different approaches to homologous recombination in humans and animals since the 1985 efforts of Smithies and Capecchi that got the Nobel Prize. And most of these since then have origins in microbial systems, including the recombinases and various nucleases. And I would say that CRISPR is likely to be a blip, just like these. That's my most modest statement that I'll make today. But nevertheless, it humbles us to see how much technology improvement there's been over the years. Here's a case study for our use of CRISPR to test 1 base pair out of 6 billion in the human genome. This happened to be a putative causative allele for Barth syndrome that has an impact, cardiomyopathy. And this 1 needle in a haystack, 1 base pair out of 6 billion, was a guanine. which we can, there are of course 3 million differences between this baby and many other people on the planet, but we could take these standard stem cell line from PGP1 and introduce a removable Cas9 CRISPR protein and guide RNAs. These are inducible, and then either do no targeting or homologous recombination, or this side product, which is non-homologous end joining. So the goal is to delete one guanine base pair, and we confirm this by whole genome sequencing of 3 different induced pluripotent stem cell clones. This was published very recently in Nature Medicine with our collaborators Bill Pooh and Kit Parker. And here's some images of where we've epigenetically reprogrammed the skin cells into stem cells, the stem cells into this exquisitely organized cardiac model with this repeating sarcomere organization in the wild type or in the non-altered control. When we change one base pair in this BTHH column, you can see a huge change in morphology as well as change in lipids and mitochondrial function and contractile function as well. And these can finish the proof that this base pair is necessary and sufficient, is actual a causal mutation. You can restore this in trans with a modified messenger RNA as shown on the right. I'm going to skip over these 2 slides so that we can save those for those of you who will be here at 10:30 for that session. We can not only interchange one base pair out of 6 billion, but we can change large sections. So we have these bacterial artificial chromosomes that are up to 150 kilobases for this particular stem cell line, and they are, we can replace entire genes or entire sections of chromosomes by having 2 crossover events that can be initiated by either 1 or 2 CRISPR events. Here are these little blue and magenta lightning rods on the left and on the right. And so this is the work of postdoctoral fellow Susan Byrne and colleagues. And it's a nice system where we can either humanize mouse or mouseize the human. And the Thy1 locus has great fluorescence activated cell sorting reagents. So we have human Thy1 fluorescent intensity on the y-axis and mouse on the x. And you can see on the far right that the lower right quadrant of this fax, you're seeing homozygous human in the starting case, and in the other ones in the upper left is homozygous mouse replacement of this entire Thy1 gene. And you can see efficiencies getting close to 12% without any selection whatsoever. This is simply by cutting on one side, in the case the left guide RNA, it's sufficient to get recombination over the entire region with high efficiency. We also occasionally observe heterozygous intermediates as well as inversions and deletions. To look into those deletions in more detail and to see the promise for larger changes over larger genomic regions, we anchored a couple of left-hand guide RNAs and 10 right-hand showing deletion, fairly flat deletion spectrum curve from about 3 to 83 kilobase pairs, and even larger rearrangements have been made, and these are not limited to deletions and inversions, but translocations as well. So epigenome engineering. So those were all using either double-strand breaks or nicases or double nicases to actually change small single base pairs or large regions. We might want to not change the genome covalently at all in terms of but to change the epigenetic state. And Kevin Esfelt and Prashant Malley, the postdocs at the Wyss Institute, introduced a number of different ways of putting in activation domains, both by fusion to the Cas9 protein of activation domains such as VP64, and we have even better ones now, or by putting them onto the RNA by a linking RNA binding protein. And you can now eliminate all the nuclease activity, which is native to the CRISPR-Cas9 protein, and now it only binds, but it will now activate transcription. And you can very easily, this is very inexpensive, about $4 per guide RNA segment. You just have to reprogram 17 to 20 base pairs, and you can pepper a region entire transcriptional with these, and you can arrange it so that if you have a very weak activator, they don't work individually, but they work in concert, and so you can get up to 30-fold activation from 10 adjacent guide RNAs, or you can get a single guide RNA that can provide up to 20,000-fold specificity. It depends on the location. We have multiple guide RNAs now from multiple organisms, and there are many more to harvest out there in the biosphere. These give us new specificities for the protein part of the specificity. Most of it's provided by the guide RNA, but 2 or more base pairs are provided by the protein. These are called PAM specificities. And we can make smaller proteins for adeno-associated virus delivery, which is quite fastidious and tight on its delivery size, and Cas9 is a big protein, so we have smaller ones now. And we can provide these orthogonal functions if you want to do activation and repression, say, in the same cell. Kevin Esfelt has now 4 of these published from these 4 different strains here with different protein PAM sites, the protein part of the recognition, where the original one had a GG dinucleotide, and then these ones have typically more complex PAM sites. The most common disease, which we all have, is aging. You can barely even see the spectrum that drops down past 110 to 120 years, but there are people out there on that spectrum, and they are supercentenarians. We would like to extend youthfulness, not just age, so that you can smoke and drink and eat sugary foods until you're 122. And one of the directions that we and our colleagues are taking on this is looking at model organisms, at long-lived organisms and long-lived people for inspiration, as well as cell biology and physiology. And we have an example of this in the mitochondrial pathway leading to nicotinamide, which Sinclair and coworkers have shown can reverse this age-related phenotype of loss of ATP and NAD plus that you see here in mice in the period from about 6 or 7 months to 22. And part of this pathway is TFAM, and so we use the CRISPR activators, not cutting now, but activators to activate this TFAM protein in this pathway to see if we could come up with a different way of enhancing NAD and ATP. And you can see that we can pepper the region as we did in the previous slide, just upstream of the transcription start site, looking for one that theoretically, or better yet empirically, like T2, has a large activation impact here. And so T2 is the best in this case, almost 50-fold activation, and then ask whether activation of that protein results in downstream physiological effects, and it does, both NAD and ATP on the left and NAD to NADH ratio on the right, the relative to the control, which has the Cas9 protein but not the guide RNA. So the presence of both the protein and the guide RNA are important for this large impact on ATP and NADH. And then finally, this we can use a variety of epigenomic modification tools to go from these standard PGP cell lines, iPS cells, to neurons with a remarkable protocol that was developed by Volker Buskampf, Nathan Lewis, and Patrick Guy, all postdocs, now professors, in 4 days. So 98% conversion of stem cells to these neurons in 4 days, and they make new synapses and are able to mature, depending on which way we nudge them, into these bipolar neurons or multipolar neurons with different properties to make these neural nets. And we've characterized these by transcription analysis and proteomics. You saw some proteomics in the previous slide. And we can identify exactly which neurotransmitters are being used in their synapses. And we can characterize their electrophysiology. In 4 days, they have this very immature spiking phenotype. And then additional 4 days, 14 days, they have a much more mature set of spikes. And finally, the last topic is how do we analyze systems like that, where we've done genome engineering and epigenome engineering. And just briefly, I want to introduce a new technology that we just published in Science called fluorescent in situ sequencing. This allows you to look beyond simple analyses of aggregates of cells or large numbers of cells that are assumed to be homogeneous. We find that in reprogramming and in natural tissues, there's a great deal of heterogeneity that's very biologically meaningful. So this allows you to quantitate by individual counting of every RNA molecule having its own sequence tag. So you do next-gen sequencing, but instead of randomizing the RNAs onto a slide, you retain their 3D coordinates. And I'm not going to go through the details of this, but I urge you to take a look at this if you're interested in analyzing the genomic and epigenomic effects that you're taking on stem cells. We are aiming for more than 2 reads per molecule, and we can do this by stratification, super resolution, and serial sections. And here's an example of this instrument showing excellent quality for those of you who do next-gen sequencing or see the quality. Here are these tetrahedron where there's almost no overlap between the 4 bases A, C, G, and T. And here's some data from a bone osteosarcoma and a variety of other cancers that have been done by collaborators in the Beth Israel Deaconess and the Pandolfi lab. So I'm going to end there. A little bit ahead of schedule, get us back on, but time for questions if there are any. No questions, so we'll just use the time wisely.